In this video, we will discuss about this ITGA3 and ITGB1 role in the pancreatic cancer progressions. So far, we have understood that these selectins are overexpressed in the tumor microenvironment of the pancreatic cancer cells, and the glycoproteins that interact with the selectins and that are also found to be overexpressed in the pancreatic cancer cells are these ITGA3 and ITGB1. These things we have discussed in our previous videos, where we have understood the results that are presented before this figure in the same paper. And now they wanted to check whether this ITGA3 and ITGB1 helps in this cancer cell progressions. So the hypothesis is that this ITGA3 and ITGB1 helps in tumor immune suppressions, and thereby they help the pancreatic cancer cells to move to the next stage and metastasize and spread across different parts of the body. So initially what they did is they started to check whether this ITGA3 and ITGB1 are overexpressed in the pancreatic tissues compared to the normal tissues or not. So for that they checked this ITGA3 and ITGB1 expressions, and for the control they use CD24. So CD24 expression levels are not changed, whereas the ITGA3 and ITGB1 expression levels are significantly increased, indicating that they are specifically increased for the case of pancreatic cells or pancreatic tissues compared to the normal tissues. And thereafter, they once identifying that ITGA3 and ITGB1 high expression levels are the markers for these pancreatic cancer cells, they wanted to check from the publicly available data that whether there is a correlations between ITGA3 and ITGB1 with the survival probability or percentage survival rate. So for CD24, as they used it as a control, they do not see any significant changes with high CD24 and low CD24 in terms of the percent survival rate. But when they check it for the ITGA3, they are able to see that with high ITGA3, the patient percent survival is low compared to the low ITGA3, where the patient survival rate are high. And similar things they have find out for ITGB1, where the low ITGB1 will result in the high percent survival rate, whereas high ITGB1 results in low percent survival rate. So once the confirming from the publicly available data, that is these data they have used from the Cancer Genome Atlas or TGCA, which is a well-known publicly available data for the cancers. And after that, they wanted to check the correlations of this ITGA3 and ITGB1 with the pancreatic cancer progressions. So for that, for the CD24, they are able to see a inverse correlation that there is a decrease in the CD24 as the pancreatic cancer cell progress from this incipient stage to the metastatic stage. However, for the ITGA3 and ITGA3, they are able to see that there is an increase in the ITGA3 expressions as the pancreatic cancer cell moves from borderline to resectable to locally advanced and finally to the metastatic stage. So indicating that as the pancreatic cancer cell progresses or become more aggressive, the role by the ITGA3 and ITGB1 is also increased, and they continuously helps in this immune suppressions. And similar things or similar results they have observed for ITGB1, though it does not seem visible, but they mention in their text that both ITGA3 and ITGB1 has direct correlations with the pancreatic cancer cell progressions. So far they have made the three different points that selectins are overexpressed in the tumor microenvironment, and the glycoproteins that plays a role in the that can interact with the selectins receptors are the ITGA3 and ITGB1, which are also found to be overexpressed on the pancreatic cancer cells and not overexpressed on the normal pancreatic cells. So all these things they have able to conclude till now in this paper. And the other things that I have mentioned, that is glycoproteins, the ITGA3 and ITGB1 expressions, or ITGA3 and ITGB1 are the glycoproteins for selectins, they find out using SPR, that is surface plasmon response, surface plasmon response. So from that they are able to find out that ITGA3 and ITGB1s are the one that interact, are the glycoproteins that can interact with the lectins. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.